Hello and welcome everybody to my channel Omni Bio Entrance, and we will be discussing CUET entrance exam question paper for the subject Botany conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want our previous year question papers of various CUET entrance exams, you can go to my channel Omni Bio Entrance. In the playlist C for CUET, and there you will find. Coming to the first question, reserpine is obtained from the roots of Ravolfia serpentina. Datura inoxia, it produces tropane alkaloids such as scopolamine and hypocyamine. So, and Withania somnifera, it produces withanaloids, not reserpine, and digitalis purpurea, it contains cardiac glycosides such as digoxin and digitoxin. So they have asked for reserpine, it is found in Ravolfia serpentina. So again a question from economic botany, be thorough with that. Then which of the following statement is true for retting in jute? It is a kind of fermentation process. Yes, it involves the use of microorganism to break down pectin. So it is a kind of fermentation process. Yes. It employs the action of bacteria on plants. Yes, that's also correct. Then in this, the cortical tissue and phloem tissue of the bark of the plants which contain free strands are decomposed to separate fiber from non-fibrous woody stem. That is what the bacteria does. So that is also correct. D, if you see, this process does not involve any microorganism. No, it involves microorganism. So A, B, C is the answer. That makes option 2 as the right answer. Then economically useful plant and the family, again from economic botany. So Corcus species, it is found in Malvaceae A2. Rosa damascena, it is found in Rosaceae B3. Piper nigrum, it is from Piperaceae C1. And Curcuma longa, it is Zingiberaceae D4. So A2 is there in 2 options. So we will see. Then B3 Rosa, again B3 is 3. Piper nigrum, it is Piperaceae C1. So that makes option 2 as the right answer. So according to Vavilov, croplands evolved from wild species in the area which shows greater diversity and richness in variation. That area is called. So it is called as the primary gene center. According to Vavilov, it is the primary gene centers. They are the geographic areas where the crop plants, wild ancestors have originated, and there you can see greater diversity and richness in the variation. Secondary gene center means the areas where the crops were introduced and where they further got developed, leading to formation of new varieties. So that is answer here is primary gene center. Then next one, match the list 1 with list 2. Numerical change in chromosome, symbolic representation. Autotetraploid. In this, the organism has 4 set of chromosomes which are derived from the same species. Auto, self. So from the same species. So it is 4x. A1. So A1 is there in 3 options. Okay. Then what is nullisomy? Nullisomy, it is a type of chromosomal abnormality where the individual does not have copies of a particular chromosomes. Okay. That is, both the copies of the chromosome are missing. So it is -2. So 2n - 2 is nullisomy. So that makes B3, that makes 2 as your answer. A1, B3. Then what is monosomy, where again a chromosomal abnormality where the individual has one copy of a particular chromosome instead of two, and it is 2n minus 1, that is monosomy, so C4. And allohexaploid, having 6 set of chromosomes, it is hexaploid, so 6 set of chromosome where each set comes from different species. So 2x1, 2x2, 2x3, so D2. So that is makes option 2 as the right answer. Then in sweet pea, the development of purple colored flowers requires the presence of two dominant genes C and R. Example CC, capital C, capital C, capital R, where either capital C or capital R or both the genes are present in homozygous state, the purple flower color cannot be produced. Okay. If capital C, small r, white is crossed with small c, capital R, white, what will be the modified dihybrid ratio of the F2 generation? So here if you see, this is a case of complementary gene interaction. That is, two or more genes, they work together or they both must be present in the plant to produce the final product. If one of the gene is absent, then it will not give rise to the exact product. So it will change the dihybrid ratio, that is your 9:3:3:1 to 9:7. Okay, so that is option 4, 9:7 is the answer. Now again explaining, so this is the cross, so capital C small r with small c capital R, these are the gametes, this is your F1 generation. Then self the F1 generation and you will get F2, these are the gametes, these are the other 4 gametes. The ones which I have underlined, they are all purple because in them both capital C and capital R genes are present. But those, and they are 9 in number, 1, 2, 3, 4, 5, 6, 7, 8, 9. Those where only one of the dominant gene is present, not both. That is this case, this, all will be white, they will not be purple. Okay, that is the case of complementary gene interaction, and the ratio that you get is 9:7. Then again assertion, reason, penetrance and expressivity. Okay, penetrance and expressivity are terms which can be used interchangeably. No, that is not correct. Penetrance, it refers to the percentage of individuals with a particular genotype who expresses the associated phenotype. It is to the percentage of individuals, not just one individual. It is the percentage of individuals who are having a particular genotype who express the associated phenotype. That is, the phenotype associated with this particular genotype is expressed. That is the penetrance. And expressivity, the degree to which a genotype is expressed as a phenotype within an individual. Okay, that is expressivity. So it can be like some individuals who have this genotype may show mild symptoms of that particular in the phenotype. No, okay, and some may show severe symptoms of the phenotype. That is expressivity. And penetrance, it is the percentage of individuals with a particular genotype who express the phenotype, that is penetrance. So here A is not correct, but R is correct. So that makes answer 4 as right. A is not correct, R is correct. Then next one. Calculate the degree of freedom if the given data set represent a dihybrid cross. Now you know in a dihybrid cross you got 9 yellow round, 3 yellow wrinkled, 3 green round and 1 green wrinkled. When you did Mendelian studies, that time you got 9:3:3:1. So there are 4 phenotypes in a dihybrid cross. 1, 2, 3 and 4. They have asked to calculate degree of freedom. So degree of freedom is number of phenotypic classes minus 1. So here how many phenotypes? 4 phenotypes. So 4 minus 1, that will give you answer as 3. So that is option 1 is the answer. The transfer of DNA from one bacterial strain to another by virus, that is important, and the subsequent recombination between the chromosomes of the recipient cell and the introduced DNA. Okay. Now what they have asked in the question is, your key word is here is by virus. Transfer of DNA from one bacterial strain to another by a virus, that is the key word, by a virus. Okay, so and what happens after the transfer of the DNA? The introduced DNA, it will recombine with the chromosome of the recipient cell. Okay, that is what is they have told in the question. So you have to see for the keyword, that is your by a virus. Transfer of DNA by a virus, that is called as transduction. Whereas in transformation, that is the bacteria will uptake free DNA molecule from the environment. Conjugation is the process where bacteria transfer genetic material from one cell to the another through a specialized structure called as the conjugation pilus. So where virus is involved in the transfer of DNA, it is transduction. Then next question. In the polyacrylamide gel electrophoresis, what is the role of APS, that is ammonium persulfate? So the various components of a polyacrylamide gel is that it will have acrylamide, which is the gel matrix, then bisacrylamide that will cross-link the acrylamide, then TEMED is there that will act as a catalyst to accelerate the polymerization of acrylamide. Then there is ammonium persulfate, which is an oxidizing agent that is used with TEMED to catalyze the polymerization of acrylamide and bisacrylamide. So 3 is the answer. It is an oxidizing agent. And what does this polyacrylamide gel electrophoresis used for? It is used to separate proteins based on their size. Okay, usually it is used for protein separation based on their size. Then next one, what is the correct sequence of steps which take place in polymerase chain reaction? So in a polymerase chain reaction, the first will be denaturation of your double-stranded DNA. Then comes annealing, where the primers will anneal to the DNA. Then comes extension. The Taq polymerase will come and act, and it will extend the primers, resulting in the synthesis of double-stranded DNA. So the answer here is denaturation, annealing and extension. 2 is the answer. Then next one. During the germination of cereal seeds, what is the role of alpha amylase present in the alluron layer? So amylase, it is an enzyme. It directly gets converted into sugars. That is not correct. It is a reserve food material. Amylase is an enzyme. So it hydrolyzes the endosperm starch into sugars which provide the energy for the growth of roots and shoots. That is more apt. Amylase is responsible for seed dormancy. That's also not correct. Then next one. Which disease is a severe form of protein energy malnutrition which results when a person does not consume enough protein and calories? So it is marasmus. That is the answer. Lathyrusm, it is a neurological disorder caused by eating Lathyrus sativus. Then favism, it is a genetic disorder due to non-ingestion of or deficiency. Sorry, 1 minute. Okay, favism, it is result in a deficiency of glucose 6-phosphate dehydrogenase after ingestion of fava beans. That is favism. Then night blindness, it is caused by deficiency of vitamin A. So the answer here is marasmus is the answer. Then next one. There are different possible glucuronides that can be used as substrate for beta-glucuronidase. Okay. Gus reporter system. Depending on the type of detection needed, the most common substrate for Gus histochemical staining is, substrate is called X-gluc and the full form is 5-bromo-4-chloro-3-indoyl glucuronide. Answer is option 3. Here they have asked what is the substrate for Gus. That is the question. Then next one, which of the following cell organelles is regarded as semi-autonomous? So it is the chloroplast because it is having its own DNA which it can replicate independently, even though it depends on the cell for a few of its functions. So chloroplast is called as semi-autonomous. Even mitochondria is also regarded as semi-autonomous. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want previous year question papers of various CUET entrance exam, you can go to my channel Omni Bio Entrance. In there, the channel playlist, C for CUET and there you will find. Thank you.